This video is on learning targets 3 and 4. Unit 3, cell parts and functions. So in order to talk about cell parts, we have to talk first about an organelle and what an organelle is. Organelles are specialized structures inside cells that perform specialized functions or specific functions. Prokaryotic cells are limited to just a few organelles, whereas eukaryotic cells may contain many different kinds of organelles, and with most of them being surrounded by some sort of membrane. And so we're going to talk about the different kinds of organelles as we go. A couple other words to be familiar with before we get talking about the individual parts. Cytoplasm, this is the area between the plasma membrane and the nucleus. So if you look at this picture, here's the nucleus right here, and here's the plasma membrane. And so the cytoplasm is all this business in here. All of the organelles along with the fluid that is in there as well. The fluid is actually called cytosol, C-Y-T-O-S-O-L, cytosol. This is the liquid portion of the cytoplasm. And again, this contains not only the organelles, but lots of nutrients, lots of minerals essential to the cell. This is also where all the reactions take place. We're going to talk about a couple of major reactions that take place inside the cell. And a lot of you are going to say, well, you came up with a hydrogen ion. Where did it come from? Well, these kinds of things are floating around in the cell. The cell is constantly breaking things down and using those pieces. So we're going to get into that more when we talk about photosynthesis and cellular respiration. So next is the nucleus. The nucleus is the control center of the cell. The nucleus controls the cell because it houses DNA, and DNA codes for proteins, meaning that the information on DNA is information on how to make proteins, and proteins are essential for almost every cellular process. So when we say that the nucleus is the control center of the cell, all the information needed to control the cell is located inside the nucleus. The nucleus is surrounded by a nuclear envelope. You can see that here. It's this double-layered structure around the nucleus, protects the DNA, and it keeps the reactions inside the nucleus inside and outside outside. It separates the nucleus from the rest of the cell. You can see that there are tiny little pores all over the nuclear envelope. And these nuclear pores are tiny holes, and they allow the passage of materials, particularly allowing the passage of something called messenger RNA, which we'll talk about in a later unit. The nucleolus is this center unit here, this little dark purple patch. You can see it, the nucleolus. This is a dense area inside the nucleus. This is where ribosomes are made. And then there is the chromatin. Now, what is chromatin? Chromatin is the material inside the nucleus that contains the DNA. And it's also lots of proteins kind of wrapped up in keeping the DNA orderly. But it looks like, the best way to think about it is a big plate of spaghetti. It looks disorganized, but it's actually very ordered, very much on purpose. But when the cell gets ready to divide, that giant plate of spaghetti, all that DNA and protein, will wind itself up onto very dense structures called chromosomes, which are what you see here. This is normally what you think of when you think of a chromosome. Look at this is the cell, they're in the middle, kind of like this. Whereas normally the cell exists in this kind of fashion with the DNA just kind of all over the place. Not random, because it's, again, very much on purpose where it is. Things are happening during that time. That's the normal life cycle of the cell. But when the cell gets ready to divide, that chromatin condenses into chromosomes. Ribosomes. This is where proteins are made. This is the site of protein synthesis. Ribosomes are composed of RNA and proteins, and they are located in one of two places. They are located out in the cytoplasm. They're free ribosomes. These are ones that aren't attached to anything. Like you see here, there's a couple of examples of these, free ribosomes. And then there are attached ribosomes as well, and these ribosomes are attached to the endoplasmic reticulum, which we'll talk about now. So the endoplasmic reticulum is a part of what's called the endomembrane system. So endo means inside, membrane, system. So this is a system of structures inside your cells that are there to help transport proteins in and out of the body. And so the endoplasmic reticulum, its purpose then, part of its purpose, it's both the smooth ER, which you see here, nice and smooth. They don't have any ribosomes on them. And the rough ER, called rough because you can see it's dotted with all the ribosomes. The rough ER is covered with ribosomes, and why do you think it's covered with ribosomes? Well, this is where protein synthesis is going to occur. Now, not all protein synthesis, some of it takes place out in the cytoplasm, but a lot of protein synthesis happens right here on the endoplasmic reticulum, and the proteins that are made in this way will get shipped out of the cell. The smooth ER is the site of membrane lipid synthesis. So remember, the cell membrane is made of lots of phospholipids. This is where they're made, in the smooth ER. And the smooth ER is also a site of cell detoxification. So the Golgi apparatus is next, or sometimes you'll see it called the Golgi body. The Golgi apparatus will receive proteins from the rough endoplasmic reticulum via a structure called a vesicle. And a vesicle is just a storage sac, and you can see these little secretory vesicles up here, and they're like a little sac made of membrane, and they have a protein in them. And that's how the protein gets from the ER to the Golgi apparatus. Now, once they're in the Golgi apparatus will then modify the protein so that it will know where to go. Much like a post office, if you take a package to the post office will put a shipping label on it. They'll put like a postage stamp or something like that on it, and then send it on its way. And that shipping label tells the carrier where to take that package, where it should be going next. And so the Golgi body basically puts like a shipping label on the proteins so that they know where they're going in the body. You know, your pancreas produces insulin, which is a protein, and that insulin controls blood sugar all over the body. And so how does the insulin know where to go? The Golgi body puts a stamp on it, or it's like a little carbohydrate, and it tells it where to go. And so this picture kind of shows you from protein production to shipment out of the cell. See the rough ER there on the left? The proteins are made in the rough ER, then they're shipped via vesicle to the Golgi. In the Golgi, there's a tag put on it, tells it where it goes, and then it goes to the cell membrane where it is shipped out of the cell via a process called exocytosis, which we'll talk more about later. And it can also come in this way. Endocytosis is where it comes in. It goes into the Golgi body and is processed sometimes when the cell receives proteins as well. Lysosomes. Lysosomes are a special structure inside cells that break down old materials, break down materials that are foreign to the cell, like a bacteria or something like that, recycles old cell parts. Inside the lysosome, there are digestive enzymes that will break down those old cell parts so that they can be used by other things. Remember we talked about the different reactions that go on. You have a starch, you can pull it apart and get the individual sugars. Well, if you can just imagine a bacteria cell, that your cell doesn't, receives as foreign, it eats that bacteria cell, and it can use all those parts for our own cells because it's just chemicals at the end of the day. All right, a vacuole. These are mainly found in plant cells. These are used to store water and other materials like sugar. Plant cells have a large central vacuole, like you see here in this picture. It gives the plant cell structure as well. The mitochondria. Mitochondria are found in animal cells and in plant cells. Mitochondria are found in plant cells. These are the site of cellular respiration. You'll get enough cellular respiration later. But this creates all the energy that is used by the cell, or most all the energy. You can see that it's a double membrane. And mitochondria actually have their own DNA and ribosomes, which is pretty interesting. Chloroplasts are found in plant cells. These are the site of photosynthesis. They convert sunlight and carbon dioxide into high energy sugars like glucose. Again, only found in plant cells. We will talk a lot more about photosynthesis in the coming days, so I don't want to spend too long on that, or the anatomy of a chloroplast for that matter. But chloroplasts are interesting too in that they also have their own ribosomes and DNA. The cytoskeleton, this gives the cell structure and support. It aids in the movement of the cell. It aids in the movement of materials inside the cell, as long as, or also just moving the entire cell. There are a couple of kinds of cytoskeleton. There are microfilaments. Microfilaments are thread-like structures created from a protein called actin. And this gives the cell structure and flexibility used to move the cells around their environment and so forth. Actin and myosin is another protein that are found in muscle cells that are used to contract your muscle cells. Microtubules, these are straw-like in that they're kind of a hollow structure. They basically kind of act like tow ropes in the cell in that they move materials around inside the cell. During cell division, they attach to your chromosomes and pull them to either end of the cell. You can see that here, and they're made of protein. You don't have to get too detailed about this picture, other than to know that they're kind of a flexible structure. The centriole is a specialized microtubule, and during cell division, there's two centrioles, and they will position themselves at opposite poles of the cell, and they will begin the process of pulling the cell apart so that it can divide equally. A couple other structures that are made of microtubules. One of them is called cilia. Cilia is hair-like projections that move the cell around. So these are, they look like a shag carpet or something like that, but they actually kind of move around in a wave motion in order to move cells, like a cell, like a living organism, like a paramecium is an example of a little protist. It has these all over its body, allows it to move. But you also have cells inside your body that have these cilia. A flagella is another example of a microtubule. It's a long whip-like structure that can move the cell, almost like a propeller or something, or a paddle. Bacteria cells have flagella. Sperm cells also have a flagellum in order to propel them to their destination. And lastly, it's something called pseudopodia. Pseudo means false, and pod means foot. So this is a false foot. This picture doesn't really do a good justice, so I will kind of demonstrate here. So let's say you have this little guy here, and this is an amoeba. and the amoeba wants to move here. Well, the way that it will do that is it will actually project out part of its body in order to kind of pull the rest along. And this is called a pseudopod. And then it will move along until it's here. Maybe it wants to go this place next time, so it will kind of stretch out its body to pull itself along. And so you kind of see that here, whereas the cell changes shape and kind of grows this false limb in order to propel itself forward.